Thank you so much. And thanks for putting this together. It's fantastic. It was great to hear about the natural history study, and I'm really excited to read the paper. I hope you can hear me okay and see the screen. It looks like everything is going well. So today I'll be talking about interventional genetics for ZARD and specifically what that means. And I'm going to address it a little bit more from a foundational perspective. I'm not going to go too much into maybe the actual scientific findings from my lab, but maybe describing a little bit more of the sort of the basics that are underlying this process and how we're actually trying to make some of these genetic disorders actionable. And we had a already fantastic, I think, introduction to some of these things here and a good segue. But I'm just going to start with really going back to maybe some of that high school biology, just to give everybody a little primer here, where every cell in the human body has a nucleus, and that nucleus has DNA in it. That DNA gets transcribed into messenger RNA, and that messenger RNA is then read into a protein. And the protein is just a sequence of these little amino acids here that are made from these codons of messenger RNA. And as an example, ZC4H2 has 224, canonically speaking, of these amino acids back to back. And then also another important feature in specifically for most genes, there are two alleles around, depending on, for ZZ4H2, this is an X-linked gene. If you're male, you only have one allele in this case, but a female would have two alleles. So one from mom, one from dad. In a normal situation, these would both make proteins. And as we're talking a little bit about some of these genetic findings that you get maybe from genetic counseling after doing genetic screens, what sometimes happens is a mutation just sneaks in there. So where normally there would be a G here, now all of a sudden this G gets turned into an A. So a guanosine gets turned into an adenosine. So when you get your genetic counseling report, it will say something like C, which means this refers to the DNA, that there's normally a G that is now an A. Likewise, on a protein level, on what that would look like, that now changes this codon that used to be a GGU on the left, GGU, that makes a glycine. Now that might be turned into an AGU, and that is a serine. So this will be called a missense mutation, where actually, as Dr. Bearden really nicely introduced it, the protein is just read wrong. There's other instances where actually this G gets turned into a stop signal that then basically makes the protein amino acid sequence just stop right there and makes a dysfunctional protein, or it might not even make any protein at all because the cell recognizes that something isn't right about this messenger RNA and just degrades it to begin with. I don't want to, there was a lot more detail, I think, from Dr. Beard's natural history study. So if you just go onto NCBI, the National Biotechnology Institute here, and look up ZZ4H2 for variants, what you actually find is a whole bunch of variants that also include actually trisomies as well as monosomies of the X chromosome. So these are some of these deletions and duplications. So this also includes Klinefelter and Turner syndrome. But then if you look at specifically mutations that are happening in ZZ4H2, such as single nucleotide variants, again, these missense or nonsense variants, there's actually a few that directly fall within the ZZ4H2 gene. Now, the question is, how can we act upon these mutations directly? And there's actually, I like to call this sometimes sci-fi because we're not quite there yet, but at the same time, also there's a massive strides that have been made actually in the last couple of years of gene therapies actually. And gene therapies are actually a thing now. While we don't have any ZZ4H2 gene therapy in the works right now, there's a lot of other rare disorders actually where people are researching it and where it's also, again, clinically approved in the US by the FDA. What's the idea here behind gene therapy? So what you can just basically do is you grab the DNA sequence and stick it in some sort of envelope and deliver it to the patient, where the patient would normally have, again, the same mutation that I introduced earlier. Now you reintroduce a functional copy that is lacking in cells of the body. We actually do this in my lab directly via making adeno-associated virus, which is considered the gold standard right now for gene therapy. And this is something that is, again, used in the clinic. I think there's actually more than 5 FDA-approved drugs out there right now that to some degree are actually curative for the indications that they're treating. So in this case, the DNA sequence gets packaged into these viral vectors that then gets infused into the patient. And in our lab, we have a capability to generate those. It's a very standard procedure. The nice thing about these adeno-associated viruses is they can actually target cells of the central nervous system. So there's this thing called the blood-brain barrier, which makes therapeutics that go to the central nervous system just very challenging. But depending on kind of what you put on the outside of these capsids, we call them, so that make up the AV, they allow the viral particles then to travel across the blood-brain barrier in some instances, where maybe the route of delivery of such a drug is not as invasive as it could be by, for instance, a direct intracranial injection, where you would inject the virus directly into the brain. And this is just a little cartoon here that shows basically what the virus does. It recognizes a cell, a surface receptor on the cell, and then it releases its cargo through different steps. And with the end goal to make messenger RNA, again, similar to what you saw in the earlier cartoon, that then gets translated into proteins, and these will be transgenic proteins. So this would be a gene supplementation strategy, again, where you would reintroduce a novel or a protein that is defective in the original cells, and thereby hopefully improve some of the functionality of the cell. So again, there has been fantastic strides being made with this, and AV is a fantastic delivery modality for genetic interventions. There are a couple downsides with AAV. One of them actually is, generally speaking, or with gene therapy as a whole, there's a lot of genes that are actually sensitive to dosage, we call that. So if you have too little, which you can see on the left side, that's pretty bad. And then if you have too much, that could also be detrimental. So for ZC4H2 specifically, I think a natural history study and doing this very important work will help us answer if there's any sort of phenotypic correlation to having multiple copies around. But for instance, for those individuals that only have a functional, or that actually in females specifically have no functional copy in 50% of the cells, bringing it then back up maybe to a single copy is very, very important and requires maybe some vector design. So that what we actually go to is actually using the endogenous mechanisms, so the cell intrinsic mechanisms, to regulate the expression of genes. And we had some fantastic segues to that as well. So what happens in females, actually, in female somatic cells, so every cells that are not the sperm and the egg, basically, is there's in a very early stage in development, both of the X chromosomes are activated. And then very early in development, this long non-coding RNA called exist is being transcribed from the to be inactivated X chromosome. The exist long non-coding RNA then paints, we call it paints, the inactive X chromosome. And this creates what you can actually detect under a microscope to some degree, which would be this bar body. And the bar body now contains the allele that is silenced. The allele being the copy of the gene, again, that is either from the mom or the dad, while the other copy is on the active X chromosome. And this process is completely random, right? There might be some level of skewing, that you have maybe more skewing towards the paternal allele sometimes versus the maternal allele, or vice versa. But in general, this process is random. In an individual that has two, I'm just going to call them wild type copies of the gene, this means that every cell expresses either allele of ZC4H2. In an individual that has a mutation, that means that in 50% of cells, these express the wild type allele, and 50% of cells express the mutant allele. In those cells that actually have the mutant allele silenced, so again, in a situation where, the wild type allele, where the wild type allele is the one that's off in this bar body here, and the mutant allele is on. Yeah, but is there a question? Okay. Whenever that wild type allele is silenced here, one could imagine going back in there and turning back on that wild type allele from the silenced X chromosome. And naturally, actually, the X chromosome is kind of interesting because it already has genes that, despite being on the inactive X chromosome, they are still being expressed. Those are called escape genes. So the goal of my lab and my collaborator's lab, Dr. Fink, is actually going into these cells here and trying to reactivate the inactive wild-type copy of ZC4H2, thereby hopefully rescuing whatever the deficit is that comes from having the mutant allele being active. So again, what this would look like using our cartoon from the beginning in an individual that has two healthy copies, either healthy allele is on. In an individual that has a mutant, in 50% of cells, the wild-type allele is on. and in 50% of cells, the mutant allele is on, that then makes mutant messenger RNA in 50% of the cells. The goal here is to go into those cells that have the wild type allele off, is to turning back on this wild type allele and causing a gene reactivation. And so we do this testing in disease models. So we're able to work with patient cells, so either patient lymphoblasts or fibroblasts, but also what we were able to obtain through Corel, through the foundation, was induced pluripotent stem cells. So these are actually cells that have been, in a way, transformed in the lab to be able to turn into every cell of the body, minus the germ cells again. We can, what we call, differentiate them. So we specialize them to become neurons in a dish. So they go through different stages of this differentiation process, where first we go and actually create neuroprogenitor cells. And then these neuroprogenitor cells can then also get turned into neurons in a 2D culture, so where they're just a flat layer of cells. But what the lab is also really interested in is actually creating organoid models from these as well. Where these are actually representing what we would consider a mini brain in a way. So where they actually are self-assembled structures that contain different neuronal populations, as well as other support cells of the brain. What this work actually is done by Yumira, who's also on the call. She's a graduate student that I was able to take onto this project with the funds that I received. And she's doing a fantastic job. And what you can see here actually is a stain for specific markers of these differentiation processes. So that you can look at, for instance, early markers of development, such as these nestin or PAX6 markers. And then you can do that compared to a control cell line, whatever that might be. So it might be a healthy parental control, or perhaps a corrected control where the mutation has been fixed. And then you can start looking at, are there any differences in this process of differentiation from using these premature cells going into more mature neurons? And can we actually link that back to some of the disease phenotypes that are being observed, such as if there is an imbalance in excitatory versus inhibitory neurons that are being produced? Could that be somehow linked back to maybe seizures that are being observed? And then also, can you apply some of these reactivation modalities to fix these underlying cellular phenotypes? What we are also, one last thing that I want to highlight here is, as being a lab in California, we have the California Institute for Regenerative Medicine, which is a fantastic pipeline that actually allows us to go from a discovery grant, which you can see here on the beginning of the Golden Gate Bridge, all the way down to a clinical grant. So they fund all the way through clinical trials, phase 2 clinical trials here directly in California. As long as there is a patient population that is present in the state of California, you're eligible for this application. And generally speaking, the way our lab has done this in the past is we use pilot awards, such as the ones that we have received through the ZC4H2 Research Foundation, to start generating these sort of seed data sets that we can then hopefully use for bigger applications that then allow us to go through this pipeline. And I'm really happy to talk more about this, but specifically what we're working on in the lab currently is how can we actually turn these reactivation modalities into something that is actually a injectable biologic, so something that we can use to deliver to directly cell models, as well as ideally in the future also mouse models of disease. And then that allows us to study, can we rescue any of the aspects that are being observed in ZC4H2 associated rare disorders? And with that, I'm happy to take any questions or go also into more detail of some of the maybe findings in the lab.
Wow, that was absolutely fascinating. I think it's the first time that we're doing this type of research, and so incredible, interesting, especially the fact that there is a pipeline to the future possible if we continue with it, which is from, staying in your lab to making probably a biological or something that will hopefully help our children in some ways. Any question? Well, thank you. First of all, thank you, Julian, for everything that you guys do. It's just wanted to, for all the people present here, that what Julian is doing in his lab is very unique. There's not many labs in the world doing this or that have succeeded in doing this. Hence his his popularity in the subject for all the X related conditions. And we're very, very lucky that you and your team wanted to work with us in this type of research that has huge potential for incredible outcomes in the future.
Of course. I thank you guys for putting all this together, and also for allowing me to get to work on this too. And Yumira, who's also on the call, by the way.
Oh, well, nice to meet you, and thank you all so much for your work.
This is David. I just want to say that was an amazing talk. That was just really that was such great research. I'm so impressed by what you're doing in your lab.
Thank you so much.